It's 10 o'clock. Welcome to Kraków, Life Science Kraków. We are at the beginning of the third session during the Life Science Open Space Online Week planned for this year. It's my pleasure to welcome you to the health technologies session, which will be led by, organized and will be led by our partners from Europe. So we will travel today and tomorrow quite a lot in different places, Before giving the floor to host Damday, who is the today's leader, I would like to stress and put your attention to the ClusterXchange Program we are all partnering in. This information about the ClusterXchange Program you will find on our websites and of course on the website related to this session. But here is the why we are all together, because we agreed to create, develop and introduce the collaboration strategy between our clusters. The strategy which will create European partnerships for personalized medicine. So everything what you will be hearing today and tomorrow in the technologies for health sessions is somehow related to this subject. These partners are, of course, Atlanopol from France, BioVin from Wallonia in Belgium, and BioMunich, and Life Science Kraków. So welcome to Kraków. The Kraków, you can feel yourself as you are in Kraków. Kraków is your host and our great partner, Kraków City. And I'll give the floor to host Damday.
Thank you very much, Kazimierz. It's a big pleasure, and I'm very happy that I can start the 2 days program of Life Science Open Space Online Week 2020. It's a special pleasure for me because it's happening in Kraków. This is going back to my roots somehow, because my mother was born in Kraków 102 years ago. Unfortunately, I do not speak a lot of Polish, but just nie rozumiem po polsku and dzień dobry and bardzo dobrze and things like that. So let's stick with the English. And I will try to give you in the next minutes an overview about the tools of life sciences in Munich. And I hope you can see my screen. Yeah. It will be a short introduction to what's coming, let's say, which will be the exciting talks where we will present to you 4 examples from Bavaria, from Munich, and 1 example which is in the greater Munich area, which is the health cluster of Wallonia. Thank you, Marc Duchamp, that you joined us, because you are one of our partners in this European C4BIG program. Thank you very much. So why did we choose this topic? Why was this topic selected? I think it's not probably clear to everybody that the tools for life sciences make up, let's say, the progress in the life science area. If we hadn't had these tools, all this progress would not have been possible. And there is also a second reason to that. As I will show you later on, let's say the Munich biotech cluster is somehow dominated by companies which work in the area of therapeutics and diagnostics development. But if they wouldn't have those tools available, and especially also around them, that if we wouldn't have this ecosystem, things wouldn't work as fast and as good as they did. Now, oops. Now my presentation does not work. It just worked. Maybe I have to do it again. Let me try again. No. This is always the problem, that this is happening when it should not happen. Now it works. Can you see that now?
Not yet.
Okay.
No, we cannot see. No, we can't see.
You can't see.
Maybe yes. I will share your presentation.
Oh, no. Let me just try once more. Okay. I have to go back. I didn't put it on the.
Very good.
Now you can see it?
Yeah.
Okay, good. Now it works. Yeah.
It works now, yes.
Okay. As you can see from here, Munich's biotechnology has a long tradition. This tradition is, when you go at classical biotechnology, it's expressed mainly at the Oktoberfest in Munich, which unfortunately did not take place this year. But this is classical biotechnology. We will not talk about this classical biotechnology and the tools how to prepare beer today. I think there are many places in the world who can do it as well as we can do it in Munich. But we will focus on the modern biotechnology which developed in the past 20, 30, more than 30 years in the greater Munich area. So let me put it this way. Everything, since Munich is a, let's say, is a biotech cluster which was created from the strong academic environment there, it goes back actually to 1984 when the so-called Munich Gene Center was created. This was then the start for the first beginnings, for the first companies to get founded in the late 80s, early 90s. And it was actually in 1989 that the first biotech company was founded in Munich area. Company which is still exists in existence, company working in the diagnostics area. Another important point in time was that 1996, Munich became one of the winners of the so-called Bioregio contest, where we not only received 50 million Deutschmarks, which corresponds to 25 million euros from the Federal Ministry of Research, but it also was the starting point for the creation of the cluster management organization BioM, which had to be founded to distribute the money and also add some more, let's say, aspects to an active cluster management. Another important point I would like to mention is that from 2010 until 2015, Munich was dedicated to turn the Munich biotech cluster into a cluster focusing on personalized medicine. We participated again in a national contest, became one of the winners, received 40 million euros from the Federal Ministry of Research, 40 million, more than 40 million from the local industry was added, and also some contribution from the Bavarian government so that we could start a program on developing personalized medicine in academia, but especially in the biotech industry. And this was very successful. And now today, I will show you the details in a minute. We have more than 240 biotech companies here located in Munich. Here you can see, let's say, the number of companies altogether. It's about 400 companies, out of which are 244 biotech companies, out of which again 185 what we call Bavarian biotech, small and medium-sized enterprises, which means these are companies that are spin-offs from local academia, from the universities, Max Planck Institutes, etc. And as you can see on the list, major focus on the development of therapeutics and diagnostics, but then on the next two places, and you can combine them, the technology platforms and the devices and reagents, a very strong aspect here. A very strong development is in the field of the tools for life sciences that have been developed. Now, when we go to employees, then in the biotech industry, we have 16,000 employees, but in the small and medium-sized enterprises, it's about 1/3 of those, 5,000. The majority is located or is occupied in bigger biotech companies like Roche Diagnostics, like Amgen, like Gilead, which have also a strong presence here in Munich. But I will focus in the coming minutes more or less on the activities and the endeavors and the results which are coming from these Bavarian biotech SMEs. Now, one of these important results is that we have a strong pipeline in drug development. So you see here the ongoing clinical studies. It's more than 130 studies that are ongoing with the same similar number of products which are currently in clinical trials. And as you can also see at the right side, there are also already 12 drugs which have been approved, which are already on the market, and one of them, or maybe even two, are blockbusters. So taking all these things together, we can add it up in the following way. In 1997, when BioM, the local cluster management, started, we started with some 30 biotech companies. Now the number has grown to more than 240. The number of employees grew from 2,500 to more than 16,000. There was no clinical trial in the beginning. Now it's more than 130, and the approved drugs are have now reached the number of 12, and we expect more in the coming months. All this was possible to strong engagement of private investors. More than 4.2 billion euros have been invested here in the local biotech industry. But what was perhaps equally important were the around 2 billion euro of investments in the infrastructure, which mainly came from the states of Bavaria. And as you have seen also from the slide, one of the slides before, BioM, the cluster development and management organization, is supporting this development since 1997. Now, what is the major focus? You have seen that, or you could guess it from the previous slides already. I already mentioned our strong dedication to personalized medicine. Now, let's say after having so to say worked in the field of personalized medicine, it was decided that we should perhaps not only focus on the topic of personalized medicine, but on the so-called P4 medicine, which also has a focus on predictive medicine and preventive medicine. And it is the participatory part of the medicine which really takes in the patients, which play a more and more important role. As already mentioned before, we had this leading edge cluster program, abbreviated with the acronym M⁴. And since 2 years, we have a small team which is coordinating a new aspect. It's called Digimed Bayern, Digimed Bavaria, where, let's say, we are coordinating a 23 million euro program, which is trying to help Bavaria transport in the area of digital medicine, focusing here especially on cardiac diseases. As already pointed out before, a very strong focus is on drug development. I already talked about that. What is very interesting is a very specific aspect in the drug development. These are the so-called ATMPs, Advanced Therapy Medical Products, which are mainly products in the field of gene therapy and cell therapy. I mentioned here already the names of those companies which are focusing on these very specific drugs. And these are, or can be, let's put it this way, can be the drugs, the medicine of the future, which are generated here by companies, by young companies, but by also, let's say, a couple of mature companies. And then the field of diagnostic development, where, let's say, fast and reliable test systems for bacterial and viral infections have been developed in the past years. That has played a very important role, especially in the past months, when there was the challenge to develop, for example, rapid SARS-CoV-2 tests. And I think some very good results were produced here from local biotech startups, like one company, which I would like to mention, GNA Biosolutions, which developed a really ultra-fast PCR test, which only takes 15 minutes. So including, let's say, the preparation of the sample and reading out the results, it takes less than 30 minutes to get a result. And it's a very reliable result, working on very small numbers, where you can detect very small numbers of viruses yet. Now, what we have developed in the past, since the network was growing and growing, we decided to develop so-called sub-networks. So, for example, there was this one subnetwork which we gave the name Impact Bavaria. It's connecting all the players from academia and industry which are working in the field of immune oncology. So there are, for example, these cell therapeutic companies, but also those companies developing antibodies, companies developing very specific diagnostic tools, especially biomarker detection, biomarker development and detection. So this is a strong network where people are competing or companies are competing, but on the other hand, they also cooperate with each other. And they have enough space also to cooperate with their academic partners. The other strong network, which, as I already mentioned, played a very important role, is the Bavarian Diagnostic Network, where we brought all those companies together which are using modern diagnostics for their test systems. And then the third network, which had, where we saw they have very strong connections to the other networks before. So we make meetings with, let's say, where the three of them get together, or where, let's say, only two partners or two of the networks meet, is the network of Tools for Life Sciences. As mentioned before, these Tools for Life Sciences play the most important role, and they are important for all the different aspects. I would still like to mention another very important aspect. Before the current crisis that we are experiencing now, we had another finance crisis in the years 2008 and 2009. And a number of our local biotech companies were really suffering a lot because it was difficult to get funding, to get financing from the investors, because the banks had their problems, and together with the banks also the venture capital companies, the private equity partners, etc. But these companies working in the tools, in the field of Tools for Life Sciences, they sort of stayed the backbone of the Munich biotech cluster, and they continued to work. They developed their reagents, their kits, their technologies, the devices, and we were very happy that they were around. So they kept the cluster during these 2 years very strongly alive. Now, when I prepared for this talk, I thought, how could I describe these Tools for Life Sciences industry which we have here in the Munich area? This is an impossible task. So I sat down and really looked at all the companies that we can have here. And it would be difficult to just present one or two of them. And that's why we were very happy when we got this offer that a small number of companies could present themselves here at this conference. In this way, you might get a very good impression of what's going on here in the Greater Munich area. So you see here all kinds of companies which work on different aspects and which are let's say, developing their own reagents and kits and tools in general. Many of those companies cooperate with the other companies which are working in the field of diagnostics development, therapeutics development. They, of course, do not only work within the cluster, but they have international global connections, and there are very strong partnerships all over the world. Another aspect which I didn't mention until now is, of course, also the field of agrobiotechnology, and this is probably playing a more and more important role as well. What I did not put on this list, which are Tools for Life Sciences as well, are those companies which use the technology of informatics, of bioinformatics. Also here in this area, we can offer you 13 bioinformatic companies who can support you in the different aspects when you are trying to resolve, let's say, results of your experiments. I just would like to mention perhaps some senior players which really play a strong part here. These are the companies EBD, which you more or less see in the middle, and it's the companies Nanion Technologies and Nanotempers. They are very strong, very grown up, very mature companies which play an important, really global role, which offer their technologies, their equipment, their devices that they have developed. And I think they are a very strong partner here in our Munich biotech industry, and we are very proud of them. Now, how can you get access to that? Of course, we would have liked to have you here in Munich, let's say a couple of weeks ago when Analytica took place. Now, this very big, well, I would say one of the biggest fairs which we have in Europe, which is taking place every other year here in Munich, where all these tools for life sciences are presented by the different companies, of course, not only the Munich ones, but they come from all over the world. They would be around. Now, some of you might have already participated in this virtual conference, which then took place, but that's of course difficult. What would be a good, let's say, way how to access the Munich biotech industry or the Bavarian biotech industry is indeed our annual report, Biotech in the Bavaria, which in this year we gave the title Responsive in Challenging Times. And here you can find it on our homepage, which is bio-m.org, and you can download it as an e-paper or you can download it as a PDF download. You will find it very easily, and I think it's the best way just to scroll through this report, and then you can find the very different aspects. I'm also very happy to accept your requests when you have a very specific questions, how we can help you, and we will be happy to lead you to the different companies which are working in this area here in Munich and in all over Bavaria. Now let me finish my talk by talking still a little bit about what BioM is doing. We are a typical biotech cluster management organization. We support the scientists when they want to create a company. We have here, let's say, developed a very specific virtual incubator program, which is called IncuLab. If you want to have more information about that, please visit our homepage. You can see a lot of information about that. And I think this is one of our major activities that we are doing here. When scientists want to come to us, when they want to turn from a normal scientist to become an entrepreneur, we really help them. We help funding those companies, not only the newly funded companies, but also those which are already around for several years. We help them, for example, to receive grants from the state funding organizations, from the federal funding organizations, or from the European Union. We were, for example, very successful in these cluster contests where we received big amounts of money, which led also to many collaborations. And then we also support those companies to get access to equity. For example, every half year, we have our so-called Bio Angels Day, where we invite, let's say, a small number of teams. Doesn't have to be companies, it can be teams just before they are founded, to present themselves, to make a pitch in front of 30 to 40 investors. Usually we do that with physical presence, but these days, unfortunately, that's not possible. We help the companies in their business development. We offer so-called Pharma Days, where we offer, let's say, to specific pharma companies that they can meet those technologies and those drug candidates, those diagnostic tools that they're interested in on one day. And of course, well, we help those companies in their business development while providing them access to the partnering events like BioEurope or BioJapan or the BioConvention in the United States. As already pointed out before, a very important aspect is to to support the infrastructure of the cluster, to support the networking activities inside. So in this way, more value can be created if these companies know of each other. For that purpose, we have these sub-networks that I introduced to you just before. And we also support those companies in their growth. We help them in their human resources management. We have a specific job market. We offer training courses, coaching courses. We have crash courses in the specific fields of biotechnology, but also in related areas. So that's what we are doing. And in this way, we could play a small, but perhaps also somehow important part in the development of the Munich Biotech Cluster. We are very proud that we could accompany, let's say, the Munich and the Bavarian biotech network here. Eventually, we want to reach our vision, which is that Bavaria should become the place for the medicine of the future. And as you can see, our mission is that we create a unique ecosystem in life sciences and digital health to enable innovation, growth, and value creation in Bavaria. So this is the end of my talk. I sort of stick to my time. Thank you very much. And let me just check that I can see if there are any questions. There is a chat. I do not see anything here. So I will check the chat once more, and if there are any questions, I can answer them later on. I would like to start now the moderation of this session after my talk, and I would like to introduce to you Dr. Moritz Völker-Albert, who is a co-founder of EpiQMax, which is a local biotech company, and the title of his presentation will be Decoding Epigenetics Towards a Truly Personalized Medicine. Moritz, you may start. Thank you.
Thank you, Mr. Domdai. I hope everybody can hear me and also see my screen.
Yes, we can see you.
Perfect. Then, yeah, good morning from my side. First of all, thank you again, Mr. Domdai. That was a very impressive introduction and keynote, and it was actually a wonderful overview of what is happening here in Martinsried, and we are slightly contributing to it. So I'm very thankful for getting the opportunity to talk and present our company's activities. And I would actually also thank very much the organizers for giving us this opportunity. Being part of a personalized medicine and health technology session, we at EpiQMax, we ask ourselves, what makes us actually biologically unique? And if you look at those two monozygotic twins that you see here on this slide, then you will realize that those twins harbor the same or very similar genetic information. Nevertheless, they will experience different diseases. They will grow to different ages. So although genetics is very similar, they will experience different lives. And so there must be something within our body that is truly unique, truly personalized, and that is not entirely based on just the genetic information. And so we at EpiQMax, we claim that epigenetics is truly personalized and makes us truly biologically unique. So epigenetics describes a mechanism by which the DNA molecule can be further modified, as you can see it in the middle of this slide. And this can either happen on the DNA itself, which is then called DNA methylation, or it can additionally happen on the so-called histones. These are the compaction blocks that are used by the cell to actually compact the large DNA molecule into the cell nucleus. Those histone molecules contain tails, as depicted on these yellow tails here. And these tails can be further modified in our cells. This is a very dynamic process, and this process adjusts or adapts to our lifestyle. As you can see, So the food, the sports, or the stresses that we are exposed to have an influence on histone modifications in every cell of our body. And it can actually adjust. This means that histone modifications can be added onto histones, and they can be also removed according to the lifestyle that we are exposed to. And it has been also shown in literature and by many years of research that histone modifications have an impact on diseases. In particular, it has been shown in the terms of diabetes, for atherosclerosis, but also majorly on cancer. So what I want to claim here on this slide is also that epigenetics is the interface between the lifestyle that we have and the disease state that we also then at one point probably face. And this makes epigenetics a very useful entity to research on. And it is, as I have said, a truly personalized discipline because it adjusts so much to what we really experience as a single person. So the pharmaceutical industry is obviously also interested in epigenetics and to understand epigenetics and use epigenetics because it's a reversible mechanism that can be used in the body. However, they face quite some problems in using this epigenetic signature in our body. And this is due to the fact that most of the technologies are based on antibodies. In general, antibodies are very useful tools, but in terms of epigenetics, they harbor problems. In particular, they harbor problems of cross-reactivity because there are so many histone modifications on a single histone tail. Antibodies have difficulties to really distinguish between single histone modifications. Secondly, overall, you need a single antibody for a single histone modification to detect. So this raises to quite high costs if you want to detect many histone modifications at once. And finally, in terms of epigenetics, antibodies are often applied in the wrong fashion. And so there is a real need in the market that technology overcomes the problems of antibodies. And this is where EpiQMux steps in. The company purpose is to enable personalized treatment by decoding epigenetic mechanisms. And we do this with a three-step process that is overall leading to a platform technology that we have established here in the past years in the Munich area. First of all, we have developed a kit that is a real device that you can use to extract histone modifications from a number of different sample sources. This can be cells, as they are mainly used in cell cultures. This can be tissue, so either biopsies or crude tissue, as well as it can be blood, mainly plasma samples. And additionally, it can be also saliva samples. With our kit, we are much quicker than any existing workflow in the market at the moment, and this is by 4 times. And secondly, we offer a very standardized process for many sample types, and therefore we actually offer a standardization of sample processment. And then after extraction of histone modifications, we apply them to a mass spectrometer. And the mass spectrometer overcomes all problems that I've shown you on the previous slide of antibodies, because in general, by analyzing many histone modifications in parallel, we can reduce the overall costs by a factor of 5. And I will show you this in a second on our examples. And secondly, we have a very sensitive and selective approach to not only identify, but also quantify histone modifications. And finally, we have developed our own software tools to analyze the quite complex data sets of mass spectrometers, but also to visualize the data and then deliver them to our customers. At the moment, this entire technology platform is a fee-for-service platform in which we receive samples from the pharmaceutical industry, from biotechs, but also from academic research institutes. We analyze them and we deliver data back to them for either drug development or for biomarker discovery. And this leads me actually to this overview slide that I want to show you today as a proof of concept, but also to convince you that this platform is very suitable for what I have claimed before. In this overview, I will show you that we have done 2 studies. The first study is analyzing free circulating histones from plasma samples for early colorectal cancer detection here on the left side, and I will go into details on the next slide. And secondly, we also looked at cellular histones from PBMCs. So these are cells that are circulating in the bloodstream, and we looked for differences between different age groups. So let me go into the free circulating histone slide. And with our partner company in Belgium, we have developed a protocol that enables us to enrich for histone proteins from crude plasma samples. And we then prepare them for mass spectrometry and analyze them with a high resolution tandem mass spectrometer. And what you can see here in this heat map, what I'm showing you here is that we analyzed not just plasma samples, but also tissue samples from CRC patients as depicted here, and the respective stroma, which is surrounding the tumor and is called the nascent adjacent tissue, and the respective plasma samples of those patients. Overall, we have analyzed several histone modifications in each row, and what you can see is that the most abundant histone modifications are also found in the plasma samples, arguing also for effect that tumor cells release histones and in general material into the bloodstream that can be then used for liquid biopsy detection and for analysis of biomarkers. So we are currently working very hard on finding the histone modifications that have a very strong correlation with respective tissue samples and the CRC tissue samples to work out better potential biomarkers. And I'm very happy to tell you that this manuscript is in preparation and will be submitted soon. So I would recommend you to stay tuned with us. Second study that I want to present to you today is a detailed analysis of histone 4 acetylation in different age groups. For this, we have collected so-called PBMCs, so monocellular cells from the bloodstream, and compared different histone modifications. If you now look on differences between young people, middle-aged people, and old people, you do not find actually a lot of differences across the overall acetylation of these histones. And this is also not the case if you look at single modifications that can occur on such a histone, depicted here on histone 4 lysine 5 acetylation. And these lysines can all be actually detected with classical antibody-based methods, but you don't see differences between the age groups. The differences become clear if you look at combinations of modifications. So this means that there are 2 acetylations on 2 different lysines, and this is the case even here where 3 lysines are acetylated. No antibody in the world will be able to recognize this pattern of modifications. And secondly, we are dealing with super low abundant modification patterns that nevertheless show a very significant difference between the old people compared to middle and young age people. And this is actually the case for quite a number of modifications, of combinations of modifications. So overall, this shows that our technology is very sensitive and helps us to detect biomarkers that we actually overlooked or would not have been able to detect with previous technologies. Overall, we have a long-standing experience in epigenetics, but also in mass spectrometry. As I have shown to you before, that by analyzing all modifications within one single run, we can reduce the cost with very effective workflows. And we are the unique provider of the analysis of combinations of histone modifications shown to you on the previous slide in the different age groups. We offer interactive dashboards and engaging data reports to our customers. And with this technology platform, we are currently the quickest service in the market for histone modification analysis. We are overall contributing and participating in a growing market. This is depicting the epigenetics worldwide market and its projection. You can see that it's roundabout growing with almost 20% per year. And the biggest part of the epigenetics market is in North America, followed by Europe. but the most growing market is actually in Asia Pacific, because epigenetic related diseases such as diabetes, but also cancer, have the most growing prevalence in the Asian Pacific market at the moment. The biggest focus within the market is on oncology, followed by non-oncology, that is then atherosclerosis and diabetes. For us, we have actually identified more than 3,000 institutions that are dependent on our service, as well as research hospitals and pharmaceutical industries. And our long-term goal is then to enter clinics and hospitals either for biomarker detection or as a companion diagnostic platform that we want to offer with our current platform technology. Let me shortly say something about the competition within this market. As I have told you in the beginning, is that epigenetics can be either read out by DNA methylation or by histomodifications. I guess you know quite a number of companies such as Grail, Illumina, QIAGEN, but also Epigenomics and others that do make a very tremendous job in the DNA methylation field, and they all use the sequencing methodology to achieve this. In contrast to this, there are companies such as Abcam or Active Motive that use antibodies for DNA methylation detection. And so they also use antibodies for histomodifications, but I hope I was able to convince you that antibodies have some difficulties. So if we come to the histomodification part in this market, then there's Active Motive, a US-based company, and EpiQMax majorly contributing to developments in this market. And therefore, we feel that we actually really fill a competitive niche in this epigenetics market in here. Our further plannings are that we want to develop and automatize our platform. Overall, we are a spin out from the LMU here in Munich, and we have established all our technologies 2 years ago. And since then, we have established the technology and we want to grow further. And by the beginning of next year, we want to automatize the workflows, enlarge in general our portfolio and publish more clinical data. And then we also want to feed our platform with much more data so that we are able to involve also artificial intelligence and smart algorithms to find combinations or single modifications that really can tell us or can distinguish different disease groups from each other. With this, we want to expand to a clinical market and also expand in a worldwide market by 2023. And we are very confident and looking forward to this endeavor. The team is composed of the founding team from the LMU. This is Victor Solis as bioinformatician in our team. There's Axel Imhoff still involved in our company, and me with a biochemistry background. Our customer base is mainly in Europe, with Belgium, Germany, Switzerland, and Denmark. We have a very strong partnership already here in Munich with 2 clinical institutions, the German Heart Center and the ISA Klinikum. And we have actually enlarged our team very recently this year with a head of technology coming from Novartis and very experienced in mass spectrometry, as well as with a lab technician, Thea. So this is EpiQMax. We decode epigenetics. We are very thankful for all support as depicted on the bottom of this page. Also to BioM, many thanks, because we are receiving quite a lot of support also from BioM. And I'm very thankful for your attention, and I'm looking forward to any questions if there are any. Thank you.
Thank you very much, Moritz, for this very clear and very informative talk. I really enjoyed it. Although we are only some rooms apart, I didn't have such a clear view of what you are doing. Now, I invite, of course, people to participate. Just send your questions through the chat. I have one question. How does the knowledge of histone modification help to get a better treatment? Can you say something about that?
Yeah, sure. I mean, as I have said, histone modifications adapt to our lifestyle. This is the one thing, but obviously they are also target for several drugs that want to actually bring, let's say, diseased histone modification status back to normal. So there are currently 9 drugs approved in the market that are targeting histone modifications, mainly histone deacetylase inhibitors. And we think by monitoring histone modifications, we can actually enable a better treatment, because then those drugs can be much better adjusted to the patient's needs and also to the outcome of the disease. Overall, histone modifications have shown that they also influence the homeostasis of a cell. So we think that epigenetics is an important mechanism to bring back cancerous cells, for example, into a healthy condition.
Okay, thank you very much. Yeah, that was especially the second part, that there are already drugs on the market, which perhaps have not been developed to be used perhaps on that purpose, but now they can be better used, they can be personalized. Yeah, that's very important. Okay, so there is one.
We have one question from Arkadiusz Sitek.
Yeah. Well, there is, it's written in Polish. I cannot read that. Maybe you can translate it. Or is that not a...
Panie Arkadiuszu?
Tak. Very nice presentation, Moritz. I just have a question, because you mentioned at the end of the presentation that you have some collaborations with clinical institutions. So to what extent are these collaborations? What kind of projects are you working on? Is it research or
yeah, I mean, it's mainly in the field of biomarker discovery. So with the German Heart Center, we obviously work on atherosclerosis, and to find also free circulating histones that can be potentially used as biomarkers for early stages of atherosclerosis, because this is also a progressive disease that is not appearing right from today on, so to say. Second, with the ISA Klinikum, we are also working on CRC patients to find also biomarkers that show a very early predictive potential to find then, to actually enable CRC detection at a much earlier time point. These are the two clinical studies and projects that we are currently involved in.
I see.
Okay.
Are you planning to use this for diagnosis for the prediction of health trajectories in the decision support in the clinic?
This is obviously something that we want to achieve. Yes, a bit of, let's say, companion diagnostics. As I have said, there are 9 drugs in the market, and what we know is that those drugs could also be better used if there would be a good companion diagnostic in the clinics with regards to epigenetic readouts. But we are not yet there. But we are working on this, and this is, I mean, that's fun to work on it.
Thank you.
Okay, as far as I can see, please correct me, there are no more questions. Moritz, thank you so much for a presentation. Thank you for answering the questions. And I will now announce the next speaker, which will be Paul Denny Goldson from CIFO. And the title of his talk is Digital Transformation from Raw Data to Visualization with Open Source. Paul.
Thank you. Can everybody hear me?
Yeah, and we can even see you. Okay.
You can see me, hear me, and can you see my my presentation?
Okay, perfect.
Yes, we can see.
Thank you. Thanks, Professor Domde. So yes, I'm going to give you an introduction to CIFO and talk about some of the services and tools that we've developed to support life sciences and organizations that are driven by science get more value from their data and how to improve their decision making and efficiency. So what we have is just some introductions about digital journeys and some of the things that people do, and then a brief intro to some of the technologies and capabilities that we apply to help people and organizations solve their digital questions and scientific questions. And those are specifically around integrating your lab infrastructure with your data infrastructures and making sure that all of these things work together very well. And the second one is tools that help people on the very first step of their journey, where they try and move from paper to an electronic or a digital aspect, where we're using robotic process automation to extract data from paper records and move that into LIMS, laboratory information management systems. So the first thing is there are a number of different things that people think of and that we work with our customers around digitalization. The first one is quite important. It's a very important aspect where data is being farified, or the farification of data. Now, FAIR, F-A-I-R, stands for making the data findable, accessible, interoperable, and reusable. And there are many organizations. There's a lot of information online about this, and it really is a kind of a pre-competitive support network and information and tools that are available to support organizations on this journey. Verification is important because it means that you can reuse your data. It means you can access the data, you know where it is, and that means that it becomes an asset rather than just being stored on a server somewhere. RPA, robotic process automation, is where we automate things that are currently done or typically done by people, and often you see a big change or a big improvement in the efficiency. And these are a couple of the aspects we'll talk about. It wouldn't be a talk about digitalization and lab informatics or manufacturing informatics without mentioning AI and machine learning, artificial intelligence, machine learning. It's the buzzword. It's kind of going up the hype curve from its kind of exposure. But there are very many examples of where machine learning, particularly where it's basically using statistics, can help augment decisions, can help augment data analysis and understanding. IoT, the Internet of Things, and edge computing have been gaining a lot of traction within the scientific community, where we're now looking at how do we connect instruments, not just in the lab, but this is where we then start to move into capturing data from external sources, from patients, from systems that are not typically connected to your network. And then moving a little bit further on into more kind of things that people can associate with is the use of wearables and the use of speech both within the laboratory, but also capturing information and distilling that information into decisionable data. And the last part are some of the underpinnings around high performance compute, the use of GPU, acceleration, and federated computing to really take advantage of big data sets and big compute infrastructures to analyze massive data sets, particularly in the areas of genomics or multi-omics. And so there's many different flavors of digitalization and what people associated with. Today's talk is going to be primarily around the robotic process automation and the data verification aspects. But as Zifo ourselves, we work across all of these areas and provide services to our customers. And the primary thing that we have to remember always is that whatever we do with the use of technology, with the use of data, with applying new ways of doing things, is are we delivering value to the scientists? Are we helping the scientists in their daily work to make better decisions, to get access to data, to make faster decisions and improve their efficiency, take away the complexity? And so when you look at the different types of questions that scientists typically have, they require different capabilities and along that digitalization journey. Typically people say, oh, can I get data from one system to another without having to copy and paste or manually transcribe that data? And so that really is a fundamental problem with data aggregation and integration and workflow orchestration. Tell me if my standards are going out of calibration is a data aggregation problem again, but then we add on the problem of being able to do the analytics and visualize that data appropriately to the user when they need it. Similar to what's the optimum instrument setting for my highest signal to noise ratio, I still have a data aggregation and an analytics problem. But as we move on to these more complicated, or what you would say simple questions, but they're very complex to deliver the answers. So a question like, have we done an experiment like this before, is a complex question, and the data structures are quite different. We need to start thinking about enterprise graph or graph technologies and machine learning to be able to look for patterns, because it's the overall pattern of the experiment that is important, not the actual precise content per se. Then we move on to bigger questions like, what's the best process for manufacturing a drug substance like my candidate, so that I can learn and apply that to what I'm doing. We then have another graph data visualization machine learning problem that we're trying to solve. And then, another one, particularly in the drug delivery and drug development processes, is there ways that I can optimize my candidate to improve specificity, lower the toxicity, etc. And again, we have data aggregation, graph data, machine learning, AI problems. And so what we see is this kind of evolution or complexity as we go through this process. And so what hinders those things? What stops us from being able to very simply show a scientist or an engineer what experiments or what tests are similar to ones before? More often than not, there is a lack of data governance within the organization. And so this stops us being able to find the data, to being able to interpret and reuse that data. And that's where the faiриfication of data is totally targeted at. The other part is the lack of technology and process governance. So everybody does what they want. Everybody tries to integrate everything that they bring in, and there's no consistency. So that increases our efficiency and technical debt as we move forward. A lot of our scientific workflows rely on unstructured data, or we capture this data in an unstructured manner. And so the metadata and the data and the workflows are quite difficult to piece together. And the one for me as a scientist that I remember, and it's still very much in play today, is there's an awful lot of manual data movement where I have to copy and paste or manually transcribe data, which really does hinder both the usability, the use, the time, but also then it makes that data unavailable typically. And so what we see from a Zifo perspective is some areas, and I'm not going to talk about all of these, but we provide services and support and consulting services on many of those areas and of the digital journey that people go on. Verification, semantic ontologies as we move into machine learning, data workflow processing, orchestration and integration, really fundamental part of a lab operation, and then a data sciences methodologies. And what we do is we provide those services to our customers in a way that they can use. So helping, if we look at one of those problems, which is how do I integrate my lab and my applications so that the data and the workflows are all orchestrated very, very simply, and I can reuse that data, saving time and improving efficiency. There's many different things that you need to think about here, where you need a kind of combination of different services where you provide understanding the lab, understanding cloud infrastructures, potentially providing blueprints, and being able to supply this information and these systems in a validated environment working in GXP or for regulatory purposes. And so The one problem with labs is that there is a massively big environment and landscape of different instruments. Here at the bottom, we've got many different instruments, all doing different things, all producing different data. And then I have many different applications above that are talking to those. So I may have a CDS, I may have a LIMS, I may have an electronic lab notebook, a product lifecycle management, a manufacturing execution system. All of these things need to talk and get data from all of these different instruments. And so we end up with a mesh, a very complicated web of integrations that are complicated to understand, and that lack consistency. So we potentially end up with data integrity issues. And so this is the first thing that we'll talk about that Zifo deliver in this context, which is a blueprint that supports the integration of all of your kind of lab infrastructure, your documents, your instruments, your instrument software, and your scientific applications, ELNs, LIMS, requesting, orchestration tools, and being able to extract that data from all of those aspects, and then using a number of open source, cloud deliverable, scalable, well proven technologies to deliver that data, to transform it into different formats, to augment the metadata so that we can reuse that data in the future. And so we extract that data from the source systems using NIFI or MyNIFI, depending on where it is. We convert that data to an intermediate data format. We use two tools, Talent, to convert that to an intermediate internal format for the different tools, different applications and instrument types. And this is where the real cool stuff starts to begin. And then leveraging Karma, which is a data transformation tool, we can augment that data semantically. So combining that data with ontologies, external ontologies, doing mappings, taxonomies, internal taxonomies, for example. And we can convert that data into many different formats, from RDF, Relational Data Framework, which is a semantic a format for supporting semantic integrations. ADF, the allotrope data format, which is very good for long-term storage of analytical data, and JSON/XML, which is kind of an easy way to consume data. And as those data then get pushed into data lake tooling, triple stores, enterprise graph technologies like Jenna, most people have an internal data mart or some kind of database that they combine their results to. And then into the JSON can go into an elastic environment, Elasticsearch environment, all of which are doing different things. So the ADF is good for long-term storage and archive. So my regulatory aspects are considered. My triple store is there to support my AI and machine learning concepts. My data mart is there for operational reporting, and my searching is for to do the interrogation and knowledge exploration using other tools, such as machine learning tools or Jupyter Notebook for the data scientists, for applying Spotfire or things similar to that called Tableau for data visualization, self-service. Shiny, for example, is another one. But the key differentiator here is we're not just pushing that data into those data stores, we're also orchestrating the communication of that data back into the lab environment by pushing that data back into the scientific applications, or updating instrument software with more information about some other results that were obtained. And so the orchestration layer there using Camunda means that we can provide a closed loop environment. That means we have much more control. We have definition and designs of where data goes, what is it doing, and we can deliver one of those key things, which is the verification of data, because we can combine data from different systems to create these ADF documents or the XML or the JSON, where the metadata is enriched by various other tools. And so that's the first aspect that we wanted to talk about. The second one is where we use robot process automation for data migration to LIMS. And so the key thing here is many of our customers and many labs still use paper, or they have a large historical use of data in their environment. And so we've designed this tool using similar technologies to be able to take that static data from Word documents or PDFs that's combined with some configuration aspects, and those are all combined into a robot process automation rules engine. That rules engine will then look at the static data and the configs and say, right, I can see all of this data in this static data, and that generates a draft specification. But that process repeats itself again and again and again, so that over time you're able to collect all the data and map all of that static data to your data landscape that you're building. And so the next step is then to take that specification for a product, for example, for a manufacturing organization, and then load that into a LIMS of your choice. So this is typically the process that people go through as they go on their digitalization journey. And the benefit of using the robot process automation and the development and the use of algorithms to analyze that static data is to generate the specification very rapidly. But the other value add is being able to take that data and using our experience, Ziffo's experience of working with over 70 different product vendors, is to deliver that data to the LIMS or the ELN or your manufacturing execution system. So typically it takes about 40 hours to do one product specification manually. One organization we're currently working with has 25,000 of those product specifications that need to be migrated into their electronic systems. So you can do the maths. That's 40 hours is probably about a week's work, and they have 25,000 of those. That's 25,000 weeks of work that they need to do. So it's an intractable problem if we just apply human resource to that. So the application of informatics and understanding and integration allows us to deliver each of those specifications in under an hour. It's fully automated, and so the power is I've taken all of that historical data and now I have it in an electronic and a digital form, which means then I can consume that data downstream. And so the key areas and things that people think about is, well, does this work for anything? Yes, any product specification or specification that contains any pieces of information can be mapped, and then that data extracted, and then those digital specifications created on and then loaded into the relevant subsystems. And so just to bring to close the element or our talk today, a little bit about Ziffo. We're 800 people strong organization. We have offices in 9 countries, the USA, in the UK, but in Germany, hence why we're talking to you today, in France and Switzerland also, and then around over in the East with Japan, India, China, and Singapore. We have many of the top biopharmaceutical companies and life science driven and scientifically driven organizations working with us as customers, and those include food and drink organizations, fast moving consumer goods, and the chemicals industry companies. And we have many customers across many different domains. And so, what we talked about here was just 2 pieces of what we do as Ziffo in the R&D in the manufacturing space, but we also have a very deep experience and domain knowledge in the clinical space, multi-omics, and then across all of those, bringing all of those different data sets together to deliver the data sciences concepts. And so that's kind of a higher level view of Zifo and what we do, based on just showing you a couple of those pieces that we provide today. So I'll bring the talk to a close. Thank you very much for your attention. Hope it was informative. And if you have any questions or want to learn more, then please feel free to contact Ingo or myself at Zifo, and we'll be happy to help. So back to Professor Domde, and any questions?
Thank you, Paul. This was a very interesting talk. I didn't know that such a world existed yet. Let me ask you one thing. Starting from what size does it make sense to make business with Zifo? Would it be, it's not probably reasonable if you are a small startup company, but for example, an institute, that would work out? Or what is the smallest biotech companies that you're working with? Let me put it.
Yeah, so we typically can get involved with very small. I think our smallest company is is an organization with 5 or 6 people, and what they want us to do is help them with their lab informatics, and we host their lab informatics for them, and we provide support services for them, and we make things work. And then as you grow, you can imagine that the complexity of that infrastructure becomes more, and they want more informatics capabilities. They want to design their processes. And so we grow, and we work with organizations that, those global brands that most people are aware of. So it really doesn't really matter how big organizations are. They typically have an informatics support problem, and we pride ourselves on our quality and our ability to deliver really cost-effective solutions.
Okay, thank you for this answer. Well, let's talk about that in more detail at a later time, because we have experienced that a number of companies also here, Munich and Bavaria, would be interested also to, let's say, to get access to AI machine learning, but nobody really knows how to do that. And maybe you can offer support in that. So everybody thinks, well, shouldn't I get involved in that? And then the question is, but how to do that? Yeah, and I think that would be very interesting to know. I've seen we've seen your big efforts there, which you probably can apply for big companies, but I'm very hopeful that it might also work for smaller ones. Okay.
It does, yes.
Thank you very much, Paul.
Thank you.
It was great, fantastic. We have to get to the next speaker, which will be a great one. I'm happy to introduce Dr. Sophia Doll from Omic Era, and what she says is, we aim to revolutionize medical diagnostics, and I'm sure you can do that. Sophia, you may start, the stage is yours.
Thank you very much. Let me just reshare my presentation here. Okay, here we go. Can you all see my screen?
Very good, thank you.
Perfect. So thank you very much, Professor Domde, also for the very nice overview about what's going on in Munich with life science. And also thanks a lot to the organizer for inviting us. We are very excited to be able to present our still young company. We actually just turned 1 this month. Our company is called Omic Era Diagnostics, and we have a very ambitious goal that also kind of goes hand in hand what Professor Domde said in his previous talk. We also believe that Bavaria should be the place for the medicine of the future. So what we want to do is basically using mass spectrometry-based proteomics, we want to profile human samples. And what you see here on this first slide is basically one of a robot that is transferring human blood into a 96-well plate to be able to be processed through our technology platform. And as you may know, there are billions of plasma samples available worldwide. And this is also due to the fact that probably, and actually hopefully every one of us already went to the doctor to get the blood taken and to get their blood levels checked. And what usually happens when you go to the medical doctor's office, they get these canules and milliliters of blood are taken out, and a few parameters will be checked by the doctor to get any information about the health status or the disease status of an individual. And these lab tests are actually extremely important because most of the clinical decisions are based on lab testing. So 77% of all the clinical decisions are made with lab tests. And among all those lab tests, the majority of them are actually based on proteins. And the proteins, those are the analytes that we are focusing on at Omega. So how are they actually measured today? So these proteins are measured based on an antibody-based platform. So usually they're measured in so-called cobas system, where one antibody measures the abundance of one protein. And antibodies are great. They work very well. They're extremely sensitive, so meaning that they can also identify very low abundant proteins. But if you have, for example, problems when the pre-analytical phase with processing the blood plasma, this can result in changes of the proteins. And then sometimes the antibody cannot recognize the epitope anymore. And this can lead to some false positives or negatives. What we aim and what we envision at Omega is basically to replace this platform with mass spectrometry. And what we aim to do here is basically work with mass spectrometers. So for those in the call who do not know what a mass spectrometer is, you can basically imagine it as a very exact scale. So we basically weigh proteins that will give us information about the identity, but also the quantity of the protein in any kind of samples. And today, due to the time restriction, I'm only going to focus on plasma. And with one measurement, instead of only getting one protein, you get hundreds and thousands of proteins in body fluid and up to 10,000 in tissues. So we really want to move towards a multi-attribute monitoring, giving you the complete picture of all the proteins that might be present in the sample. So our vision is really that these proteome scans will become a universal diagnostic tools for health and disease. We want them to be applicable to detect diseases early on, and that patients or individuals get their blood proteome profile regularly, so that they could also not only, like if you will get a blood report, you get a specific value, and if you're falling within a specific range, you're stated as healthy or diseased. But in that case, you could also be your own control. So getting like a regular proteome scale scan over the years and see whether your proteins are changing over time and try to detect diseases very early on. So we anticipate that this MS-based proteomics will become a cornerstone of precision medicine. And for that, we really want to build up the best performing technology platform with the highest capacity and precision for clinical routine. So we really envision that this technology will be transferred into the clinic, and with that, we want to improve health with proteomics. So how does our technology platform look like? This is what we do. We work with all kinds of samples. I'm just talking a lot about human plasma today, and also because this is one of our major focus of the company. We work with minimal amount of starting materials, so less than 1 microliter in that case is more than enough. But besides plasma, we can also work with all kinds of body fluids. So basically everything you can imagine we work with, going from urine, CSF, saliva, you name it. And also we work with any kind of tissue samples, so ranging from fresh frozen ones to FFPE material, and of course also cell culture. The technology platform encompasses an automated sample preparation, where the first step is basically to extract the proteins out of the samples that are then digested into peptides, and after cleanup being injected into our mass spectrometer. What you obtain is spectra for the identification and quantification of the different proteins you may identify in the samples, followed by an automated data analysis that we have developed here at Omega. So we basically offer this technology platform as a CRO, so for a fee for services. And our main clients encompasses academic collaboration partners, but also biotech companies that would like to maximize the insight of their sample by running them through our technology platform. And what we also really want to emphasize is to discover more reliable and valuable biomarkers. So we have different points that we work on and that we've optimized over the years, and this is where experience meets innovation. So all of us have many, many years of experience within the field of mass spectrometry-based proteomics. We have established very strong partnerships over the years. So we work closely with Bruker, EvoSap, but also very strongly with the clinics. And we actually also have one of our mass spectrometer being physically in the clinics. We work with very large sample cohorts because we have an automated and scalable platform. And something that's extremely important to us is the quality control of our samples. And I'm going to show an example in the next slide. We are also growing a lot on the bioinformatics side and are actually also hiring in case somebody is listening and is looking for a job. We are really expanding on that direction because of the large amount of data we're producing, and we want to get the most out of each data. So together with the technology platform, what we aim to is really to simplify this very complex technology to allow routine use in the clinic. We want to generate actionable outcomes to improve patient care and offer this high industry standard services to our clients. So as I was saying, quality control is extremely important for us. And over the years, we have developed panels, so to be sure that the quality of the samples is good enough for a biomarker discovery study. And in the next slide, I'd like to highlight why this is so important to check the quality of the samples, because in previous literature, there's been a lot of protein outliers that have been claimed as being potential biomarkers, but if one has a closer look at the data, it was actually related to the sample handling and sample quality issues. So in this volcano plot here, you actually do see a case versus control comparison. And you might think that there are quite a lot of outliers and interesting protein in this case group here. But all the proteins highlighted in blue are actually part of our platelet panel. So suggesting that somehow in this group, there's been a contamination with platelets. And if you look at these other two proteins, GP1BA, in correlation analysis, you will find that it highly correlates to the platelet panel. So suggesting that this is also most likely a false positive. But the one that remains is NRP1, and this one remains as a potential biomarker candidate as it does not show any correlation to any of our quality markers. So this really, what we want to do with that is really to help shape and really better identify potential biomarkers that are not related to any potential pre-analytical variation or any quality issue we might have with our samples. What we typically get as a plasma proteome coverage is about 300 to 500 protein identification. Something that we also do not do at Omega is depletion. So plasma is a very challenging sample where you have very high abundant proteins masking the identification of the lower abundant one. And most proteins are identified and quantified in the majority of our samples. So we have a majority of valid values in the set of proteins we identify. And this example study is a quite small cohort, we would say, for Omega. These encompasses about 200 plasma samples, but we've also had studies where we measure several thousand of samples. And this is something that's not a problem at all for us, as we have robots doing the sample preparation for us and can prepare several thousand samples per day. We are also very reproducible with precise quantification. So we obtain median whole workflow coefficient of variation of about 12%. And the majority of our proteins have CVs below 20%. So that's also something that's very important to us, that we can offer a reproducible workflow. What I was also showing on my first slide is you saw these blood cannules actually collecting milliliters of sample with a quite imposing needle. What we work at omic-Era is actually minimal amount of sample material. So just a little finger prick would actually be sufficient for us, because for our subsequent analysis, we will only use less than 1 microliter of blood plasma. And from that little information, from that little tiny amount of sample, you can actually obtain quite a lot of information. So if I would, for example, look at Professor Domdai, okay, I can say that he's a man, but I can also, that might not be obvious from his plasma sample. I don't know who this belongs to. And just by analyzing the proteome of this tiny drop of sample, I can differentiate between women and men. But I can also detect whether a woman is pregnant. I can also identify if there are different inflammation states. So we have different panels to also stage the stages of the inflammations. We can also become a lot of information about the lipid homeostasis. So with our current depth, we actually cover almost all apolipoproteins. So it gives us a lot of information about the health or disease state of the liver. We can also identify protein glycations. Something that's also very exciting is that we can identify different allele readouts. So for example, one of our colleagues found that he has a mutation in the ApoE gene. And this suggests that he has very high cholesterol level. And this is something that's quite valuable information to get to know, as you could just take statins to reduce your cholesterol levels. There's another information that one could get that maybe not everybody wants to get, is because you could also identify whether you have another ApoE variation suggesting a higher risk for developing Alzheimer's disease. And finally, we can also identify quite a lot of FDA approved biomarkers. So we cover at the current state about 80 to 100. And that's always quite valuable information to our client to also see the relevant biomarkers to be observed in blood plasma proteomes. And with that, I already come to an end of this short overview of omic-Era diagnostics. And I'd like to thank our core team. So especially my two co-founders, Philip and Sebastian, that you can also see here in the picture. We all met actually here in Munich at the Max Planck Institute, and the three of us have many years of experience in the field of mass spectrometry-based proteomics. And we're very excited also to have Max on board. He is currently leading our bioinformatics group. And as I was mentioning before, we are hiring. So if there are any bioinformatics on this call, please feel free to send your application. And we are also very happy and fortunate to have Dr. Ole Worm on board. He's a serial entrepreneur and has set up several companies before omic-Era. And his advice is always very helpful. And we're also very excited to have Professor Matthias Mann as an advisor. I also would like to thank our partners. So as mentioned, we work very closely with Bruker Daltonics. So something that's very important to us is always to remain at the forefront of the technology. So we co-develop methods, but also test alpha instruments so that we can remain at this forefront of the technology. And Evozep, who produces the HPLCs, we also have a very close connections to them, and we are always open for new partnerships to optimize our proteomic pipelines, but most of all also improve patient care. And with that, I'd like to thank you all for your attention, and I'm looking forward to any questions if they are.
Thank you, Sophia. Please accept my big applause. That is extremely impressive what you presented to us. Let me just ask you just one short question. Do you expect at some point of time that proteomics will sort of supersede the genomics area? When will it be more important and more, let's say, play a major role, a more important role than genomics, or never?
Yeah. So I guess this is always kind of the discussion, right? So will proteomics replace genomics? Will genomics replace proteomics? What we have seen in a lot of studies where we actually work with end-stage cancer patients, where they have a lot of genetic information already, because the technology has, of course, been developed many more years before proteomics and is much more advanced and can already be applied in the diagnostics field, that often this information is quite limiting. So big companies like Foundation Medicine offer amazing tests to give them more information about the state of the cancer, but often genetic information will not be enough. So what I think will be the future is really the complementing all these different technologies. Also, like Moritz presented, what EpiQMax is doing, right? I mean, not only combining the genomic, but the transcriptomic, the epigenetic, and the proteomic, and also the post-translational modification. So I really think that this multi-omic field will be extremely powerful in the future. And I think that it's really like the interplay between the different technology that will be the winner at the end of the day.
That was the Solomonic answer that I was expecting from you. Thank you very much. Yeah.
Thank you.
Indeed, this interplay between the different disciplines, which will, at the end of the day, perhaps not only proteomics, but this interplay of the different tools that will be available and will be optimized and will become better and better, will really play an important role to keep us healthy as long as possible. Thank you once more,
Hopefully,
Sophie.
yes. Thank you.
There are no further questions at the moment. Your talk was self-explanatory. No questions needed. Thank you. So let me come to the next speaker. Next speaker is Daisy Paiva from Dynamic Biosensors. And she will talk about switch sense helix, next generation modular biosensor for interaction and confirmation analysis. And Daisy and her company are located here in Martinsried. Welcome. The stage is yours.
Thank you. Thank you very much for the introduction. And thank you very much as well for the invitation for this meeting. Everyone, I think, can see my presentation, I hope.
But we see the presentation in the preview mode, but
okay.
we also see your the slide after that.
Yeah. That's better now?
That's perfect. Thank you.
Thank you so much. Yeah, so I'm here. My name is Daisy, and I'm here today to talk about the switch sense technology and our newest helix instrument. So just to give you like a brief introduction about where this instrument will be useful for the research. So in the drug discovery process, you know, that is a very long process, and you start with like millions of targets. And the initial idea is just to see if, you know, if from this huge library, some of your hits are going to be, you know, carried out to the next step or not. And the role of our technology is then to enable you to qualify these leads in the drug discovery process. So you have the switch sense technology is particularly useful, for example, in the hit to lead step, when you have, let's say, a couple of thousands leads, and you just want to see if they bind or not so if a couple of some compounds bound to a specific protein. And in the next step, when you have the lead optimization, if these compounds then change the structure of the protein, or if activates a specific enzyme. And our newest development now have been moved a bit towards in vitro cellular assays. So we are now able to also capture cells onto the biosensor surface to analyze the binding, for example, of antibodies directly to cells. So the switch sense technology is not only a high throughput technology, but it also provides you high content information biophysical analysis. So as you can see here from the presentation, our instrument can detect not only the affinity, but also the avidity, so on-off rates KD, and also conformational change and thermodynamics. Yeah, so just to give you like a brief overview about everything that we offer. So we have two types of instruments, the Helix, that is the newest one, and the DRX that used to be, that was actually our, let's say, the first generation instrument that we launched a couple of years ago. And we are the manufacturers of these instruments, not only of the instrument, but also we are the ones responsible for developing the whole software package that comes together with the instrument, and also all the consumables that are related to this technology. And on top of that, we also offer some fee-for-service or maintenance contracts, but the main core of our business is then to sell and to enable more researchers in the world to use the SwitchSense technology. Now in the next slides, I will just show you how our newest instrument looks like. So the Helix line of biosensor instruments is again powered with the SwitchSense technology. We have automatic chip loader, and the chips are automatically exchanged. And we have also advanced fluidics that enable us to quickly exchange the fluid onto the biochip, which also enable us to measure very, very fast on rates, for example, and to characterize very difficult targets. We also have an autosample with 304 and 96 well plate. This is more to enable us to be compatible with high throughput screening labs. And again, when I mention about the throughput, so of course, we have one single unit of our Helix biosensor that can measure up to 304 well plates per day, but you can also combine these different units to then achieve a higher throughput, of course. And these different units are all connected through a modular connection. So it means that you only have one computer that controls these different units, and this then enables you to have let's say, a smoother workflow. And the ligand demobilization is then carried out in vitro. So it's not directly in the instrument. We offer you some coupling kits for that, and it's just very standard and easy process. One of the differentials of the technology is also that you use a very, very low amount of sample. You use generally 5 picomole in 20 microliter of the adapter ligand conjugate per immobilization. And the cost per data point is very low. So it's roughly $2 per data point for chips and reagents. And when it comes to chips, for example, as I mentioned before, and this is a chip-based biosensor. And what we have here are DNA strands that are attached to a gold surface. So having this DNA strand for us somehow enable us to be more sensitive, because we use not only the DNA to, let's say, you can not only immobilize the DNA directly onto the chip, or you can combine a protein of interest to a DNA and then bring this to the surface. And based on the change in this fluorescence that comes from the dye that is in very close proximity to the protein, we can then measure the interactions. And again, since I mentioned that we are DNA-based, our chips are supplied with an anchor sequence that is then, a DNA sequence fixed to the gold. And what you can do is to use several different types of what you call adapter strands, that you can either have only standard protein with the dye, or you can have some DNA origami structures that allow us to bring, let's say, bigger protein complexes onto this biosensor surface. Or you can have some, let's say, some crazy structures like this Y structure that you have 2 different dyes in a Y shape. And then you can measure, for example, protein dimerization, or you could measure a binding of a bispecific antibodies to 2 different antigens simultaneously. So let's say now that I mentioned so much about how you would do an experiment, I wanted to show you like the basics of the switch sense principle and also some of the applications we have been working with. So the first, let's say, way that we look into the interaction. So the first measurement mode is called the static mode. This is a very standard one. It's just fluorescence based. So what you have here is a ligand that is connected to the DNA. And when you come in with the analyte, you would just measure the change in the fluorescence due to the binding. And we usually have, let's say, we apply this measurement mode when you want to see binding of small molecules to proteins. And here you can see that the technology covers a wide range of affinities. So you can measure some compounds binding to kinases and also some other compounds binding to human cerealbumin. So really tiny molecules, tiny compounds that are being bound to these proteins. On the other hand, you can also use for measuring the interaction of larger proteins, like an antigen-antibody interaction. And this project we have made in collaboration with Roche. And the idea here was to see how the binding would change in the presence of different pHs. So how would the pH affect the affinity of the antibody to the antigen? And you can clearly see that just by the shape in the dissociation, there is a change in this binding behavior. And when you look into this affinity plot, you can then see that, you know, at pH 5.8, you have an affinity of 2 nanomolar. And as soon as you increase the pH, so from 5.8 to 7.2, you then have you notice a decrease in the affinity. And you can also clearly see here from this representation that the on rate is basically unchanged, whereas the off rate becomes very fast in higher pHs. So here is just also a very nice example for you to see, you know, that you can use several different buffers and several different experimental conditions in the biosensor that we are quite, the experiments are still very robust. And another very nice example is, for example, when you have RNA structures directly connect to the gold, and then you can measure, for example, binding of small molecules or binding of proteins. And in this paper here, that was from a research group in Munich, they then used the switch sense data to just confirm their NMR data that the off-rate driven binding inhibition was caused by a specific mutation. So the second way that we look into the interaction is called the dynamic mode. In this case, you know, since we have here a gold surface, you can also alternate the voltage on the surface and then move from positive to negative. And since the DNA is negatively charged, this will make the DNA to switch up and down, being attracted or repelled by the gold surface. And with that, you can then see how fast these DNA nanolivers are moving. And that's why also the technology is called switch sense. And in this type of measurement mode, we use for either looking at the interaction itself of proteins or looking at the conformation of this protein. Because also if a protein has an open conformation, you know, this movement will be much slower than when the protein has a more compact conformation. And this is exactly what I wanted to show in the next two examples. For example, what we measure here is also the friction that comes from the switching. And if a given protein, you know, when it doesn't have anything bound to it, has, let's say, 0 friction, and when it's bound, when a small molecule is bound to it and cause a compaction, when we are moving this DNA up and down, you would then notice you know, a reduction in this friction, because the compaction then makes this this movement much faster. And as soon as you have, for example, a dimerization happening on the surface, that this complex gets bigger, then this movement will get slower because the friction is increased. And that's exactly what I will show here. For example, we have a stint protein that is a dimeric protein. And as soon as you're flushing CGMP, you saw that the protein was going from an open conformation to a closed one. And then you see that on the course of different measurements, the effective diameter was always in very good agreement between the measurements. And you can clearly see, that there was a change from open to closed in the presence of this small compound. And one of the, let's say, the most used technologies to detect conformational changes in proteins is SACS. And in this paper from the Scripps Institute, they compared our data with SACS data, and they could clearly see that a specific mutation of the protein was causing, exchange in this conformation. So if you compare, for example, the Y type to the different mutations, you will see that there are at least 3 that have a bigger conformation compared to the Y type, and this data was confirmed by the SACS data. And let's say in the third measurement mode, what we do is actually to combine the static mode, so just looking at the fluorescence in 2 different colors. In this way, we can, for example, follow Fred effects or follow bivalent or bispecific kinetics. So you can look at 2 different interactions simultaneously because they are reported by 2 different dyes. And in the case that I will show you here is like a protease. We have 2 different ligands, and we have only one LED on, which is in this case is the green LED. And in the first example, what we see is that when these 2 different ligands are not bound together, when you have only the LED power on, you see the emission coming only from the green dye. And as soon as you have a dimerization, you see a fret occurring. So there is an energy transferring from green to red, and you see the emission in the red channel, sorry. And we use this type of setup to look at PROTACs. I'm not sure if everyone is familiar with it, but PROTACs are proteins that interact with each other. So in this case, you always have like a protein of interest. And here we had a ligase that they are bound together. And the idea here was to screen for a small molecule that would bring these 2 molecules together and find the KD for that. And here you can see that it could nicely calculate the on and off rates in the KDs. And the most interesting thing is that also from the graphs, you can see an increase in the red channel and a decrease in the green, meaning that there is actually a Fred effect happening here. And just, let's say, to summarize, I know it was a lot of like tech related information, but my message to you was just to say that the SwitchSense technology is a comprehensive biosensor instrument that is used for detecting kinetics, affinity, and avidity, also conformational changes and thermodynamics. So we have like a wide range of applications that are very well suitable for, specifically in the DNA, RNA field, but also proteins and small molecules. There are still a lot of things that I didn't show, of course, for the sake of time, especially when it comes to nucleic acid enzyme activity, for example. But I will be also very happy to answer any questions. And also just to give you an idea about the company itself. So of course, the Dynamic Biosensors is a Munich-based company. We are a spin-off from the Technical University of Munich. We also have offices in US, so in San Carlos and Wolverine. And also we have sales partners in Lisbon, in Portugal, in Warsaw, in Poland, and in Cambridge. And we also have some other business partners in Japan and Singapore. And our customer database is comprised not only of big pharma companies, but also several well-known research institutes. In Poland, I can say we have already some customers that have our instrument and have been using it for a while. And yeah, with that, I want to leave you with a statement from one of our customers. And I hope, and also some publications. And thank you so much for your time. And if you have questions, I'll be happy to answer.
Thank you very much, Daisy. This is a very sophisticated technology, which I happen to know from its beginning because I've had the chance to accompany the switch sense technology when they were still working at the Technical University. At the moment, I do not see any questions. Yeah, people do not dare to ask. Maybe it's really tough. And I'm so happy that I'm not have to teach students anymore about new technologies. It would be difficult for me to explain. You really have to be an expert. Good luck for everything. Thank you once more. And in this way, I can immediately transfer the stage to Marc Duchamp. Marc Duchamp is the head of BioWin, the health cluster of Wallonia. So you will hear now a completely different talk. And why did we invite Marc to join us here? As you had already heard from Kazimierz in the beginning, we are partner in interesting European network and a European project named C4Big. And together with our partner at Longpol, BioWin, Krakow, and Munich, we are trying to do some interesting things in cluster development. And Marc will now tell us a little bit about BioWin and Wallonia as they have developed there in the past years also a very active, very renowned life science ecosystem, which he will introduce to us. Thank you, Marc, for joining us today.
Thank you very much, Professor Dundee. It is a pleasure for me to have the opportunity actually to present this, as you mentioned, very, very active and dynamic ecosystem. So I'm sharing my screen now. I hope that you will see it in a while. Please tell me.
Oops. Yes, perfect. Thank you.
So clearly, BioWin is quite similar to what you presented for BioHem. Is really the gateway to health biotech and medtech excellence, but in Wallonia. Wallonia is the southern region of Belgium. And I would like to start by introducing actually the Belgian context. As it was said, Belgium is really a biopharmaceutical innovation hub located in the heart of Europe, and since many years, even decades. And why is it so? Because you can see the 4 blocks actually at the bottom of the slide. We have a very strong collaboration between world-class academic and research centers. We have quite a lot of biotech in full development for the moment, and I'm sure that you will recognize some big names on the slide. But we have also large pharma companies, headquarters, R&D sites, manufacturing facilities. For instance, we have the headquarters and the R&D site manufacturing of GSK vaccines, which was originally a Belgian company, a Walloon company. We have UCB. UCB, the big stands for Belgium. We have also Pfizer, for instance, present also in Belgium, and this factory is going to produce the BioNTech COVID-19 vaccines. And Janssen Pharmaceutica, so the pharmaceutical branch of Dzięki in the north of the country, which is still based in Beerst in Flanders in Belgium, and it was created by Professor Paul Janssen. But we have also very important suppliers and innovative service providers. And I'm sure you heard about master cells in cell therapy, CDMO in cell therapy, recently bought by Catalent. It is now Catalent Biologics, but also Univercells is a lot in the news currently, and Eurogentech, also a big CDMO. And this model is really based on the collaboration and integration of university industry, but also with a very strong support of the government, whether we speak about the federal government or the regional governments, providing actually this ecosystem with a great support in terms of tax measures, incentive, and regulatory environment, positive regulatory environment. And just an example of that before moving to BioWin, Belgium is number 2 per capita in the world and in Europe for the clinical trials. Why is it so? That's clear that we have a very, very strong academic hospitals with high quality staff, very good network of hospitals, but it is not just because of that, it's because also we have a very strong support of the regulatory authorities, because in 2 weeks' time, you can get approval for a phase 1 clinical trial. That's the reason why the phase 1 unit of Pfizer is located in Belgium, in Brussels. And Novartis is doing a lot of phase 1, for instance, in Belgium as well. So you need 4 weeks to get the approval for the phase 2 and the phase 3. So meaning that in Belgium, on yearly basis, more or less, we have 1,500 clinical trials conducted from our country. And again, this is a very good example of the cooperation between companies, universities, hospitals, and regulatory agencies, without forgetting the ethical committees. Having said that, I would like now to focus on BioWin and Wallonia. So BioWin was created, actually it's younger than BioM, because BioWin was created in 2006. By the regional government in order to really develop a new industry in the region. And the mission of BioWin, which is a non-profit organization and funded 50% by the authorities, 50% privately by the members, is really to accelerate innovation. That's all about innovation all the time, you will see, and that's the reason why we are very pleased also to be connected with our partners here within the CE4 big project to have this exchange at the profit of our members and academic groups. And the vision is to continue to put Wallonia on the map, put Wallonia on the table for the high quality of its life science ecosystem. So BioWin is gathering all the stakeholders you might have in the life science ecosystem, so all the value chain, starting with the public institution and the government, the companies, large, medium, small startups, research institutes and universities, education organization, that's also important for developing talent, hospitals, easier for the clinical trials, and capital providers, whether they are public or private VCs. And the role of BioWin is really to connect this ecosystem either with other clusters, exactly what we do today and what we do within the CE4 big project, but also global markets, because we are definitely present and we support our members at the big international event like Bio International in the US, Bio Europe, or Bio Japan, for instance. The smart specialization of the Walloon region is really straightforward, because if you look at the center of the slide, you will see what is more or less related to biologics, ATMP. So we have still pharmacy and small molecules, but most of the activity is now the biopharmacy, including the cell therapy and big biomanufacturing companies. But we have also what is specific to Wallonia, radiation applied to health, either with global leader like IBA for proton therapy, or biotech companies working in the diagnostic on the diagnostic field. We have medical device and in vitro diagnostic companies, that's clear. And what is emerging in Wallonia, and strategically we are really pushing hard to develop, is data science, digital health. And we spoke and we saw a very good example during this session. We have also companies like the Analytics, OncoRadiomics, and so on, developing fast in that field. The main activities of BioWin and the cluster is beyond organizing event, and we have the BioWin Day every 2 years, which is the biggest one, but we have a series of meetings also on different topics and thematics. So we support innovation, we support the initiation of and the development of research innovation projects. And this is really the DNA of BioWin. BioWin started with this activity, but now this activity it's expanded to international business development, so support of the internationalization of the companies, and also the support of the growth of the SMEs. What does it mean? It means that now, since more than 1 year now, we are supporting also the fundraising of the companies. And skills development. For a certain period of time, BioWin was delivering skills training, which is no more the case now. What we do now is really acting as a strategic advisor for the regional authorities in order to make sure that they understand the needs of the ecosystem of today, but even more of tomorrow, and make sure that the companies developing in Wallonia or willing to develop activities in Wallonia will find the right talent for the different type of jobs you need in biotech, medical device, diagnostic companies. And so just some information about the region. So as you know, Belgium and Wallonia is really at the heart of Europe. Useless to say that the European Union and the European Commission is based in Brussels, and Wallonia is what you have in red, the region below Brussels. What is interesting to see, this is a small region with 3.6 million inhabitants. But you see the different universities, and you see the blue dots, and the blue dots are the innovative companies located in the different science park. But the good news here, it's really easy to collaborate, because if you go from one extreme to another extreme, it's just 1 hour drive. So a lot of collaboration between the 5 industrial hubs located around the 5 universities. But in reality, the biopharmaceutical industry represents more than 50,000 jobs. So more than 50,000 people working directly or indirectly in that field, with an average annual employment growth rate, and since the creation of BioWin since 2005, 2006, of 7.7% for the all type of companies. If we take the biotech, the basis is lower, you will see 13% of growth of the employment, but you will see that there is a big acceleration on the next slide. What is also very accelerated is the cumulated private capital raised by the SMEs since 2005. It's almost 3 billion euros now, but on the next slide, you will see this very huge acceleration the last 5 years. Of note, we have also very big private R&D investment on yearly basis in the region, so more or less 1 billion, 1.2 billion euros a year. And if you are Minister of the Economy of the regions, you have to really take care of this sector, because now it's even more than 27% of the total Wallonia export. The sector represents 32-33% of the total export of Wallonia, meaning that really it is the new economy and the leading economy of the region. And I mentioned also the research innovation project, so the DNA of BioWin. So since the creation of BioWin and until 2019, the cluster raised 4 consortia, 158 million euros non-dilutive money to create new IP, new startups, new assets anyway. And so I was mentioning the acceleration in the employment within the SME sector. You see on the right-hand side on the slide that since 2015, there is a big acceleration. And I can tell you that it's 2018, putting 2019 and 2020, you would see that there is really an exponential trend we have in that field for the moment. And to explain to you the type of companies we have, if we just take the 158 BioWin Walloon members by strategic domain, you see that we have 31 companies in biopharmacy or pharmacy, 18 in biomanufacturing, 8 in cell therapy, 11 in radiation applied to health. So again, cell therapy, radiation applied to health are really, really specific in the ecosystem of BioWin. So we have a critical mass. We have 42 medical devices and in vitro diagnostic companies. I said also that data science is emerging. We have already 12 companies, but we are again pushing hard to support the development. And we have 35 support and services companies helping the ecosystem to work very smoothly with a lot of competencies available. But if you look to the employment again, you see that clearly, if you take biopharmacy, biomanufacturing, and cell therapy, so biologics in general, I would say, it is almost 80% of the employment. So this is the biggest part of our activities. But it doesn't mean that the others are not important. Radiation applied to health, we have already more than 1,000 people working in that field. The same for medical device in vitro diagnostic, 250 more or less in data science, and more or less 1,000 people also in support services and companies. And I mentioned also the acceleration of the VC money raised in the ecosystem. So in the center of the slide, we have the cumulative curve since 2005 until or up to mid-September 2020. And you have a column on the right-hand side of the slide, and each slice represents a year. So you see that since 2015, I would say 65% more or less of the money was raised in the last years. So meaning again, big acceleration, big development of the SMEs, biotech, medtech companies, diagnostic companies going to market or going to phase 3 today, and attracting more and more VC money currently. And just to finish about the BioWin cluster, so what are the members of BioWin? So we have 234 members, including 10 global leaders. I mentioned GSK Vaccines, UCB, Pharma, Zoetis, which is Pfizer Animal Health, again, Catalan Biologics, IBA, Rogentech, and others. But the bulk is composed by the innovative SMEs, 187. Out of these 187, you have 158 Walloon companies. It means that we have also foreign companies on regular basis starting activities and developing activities in the ecosystem as well. But next to that, you have the 5 universities with all the research units, 400, 7 private research centers, so meaning 11,000 researchers in total, and the 20,000 hospital beds, which are really available for clinical trials. And so on top of that, you have different talent pool in creation with the different universities, and also you have also famous universities in the north of the countries, but we have also a good relationship with the north of France, les Hauts-de-France, with the region of Lille, Eurasanté, the cluster Eurasanté. So you can really find actually the talent you need for your development. And together with the supportive environment with regard to investment and tax and incentive support from the authorities, this is more or less the type of companies we have. It is not exhaustive, but you see that we have a high industry density of life science company in Wallonia. So I will be more than happy to answer your question if you have. Otherwise, like for BioM, you can find all the information on the website of BioWin, www.biowin.org. And there you can find a directory with all the companies, with a direct link with their own website. You can find the annual report, the corporate presentation, and flyers on specific topics available for downloading, available for you. That's all. Thank you for your attention, and I'm really pleased to answer any question just now. I'm stop sharing.
Thank you very much, Mark. I hope you can hear the big applause. Unfortunately, we cannot really do that. It was a fantastic presentation. I'm very much impressed about what you can do there. I would indeed, yeah, this is admirable, because you can see that here the life science sector, the biotech industry, and the pharma industry have taken the lead in Wallonia. Yeah, and I think this is also that is leading into the future, maybe, or probably better than being a leader in the automobile industry, where we do not know these days where they will lead us to. No, fantastic. Thank you so much. At the moment, I do not see any questions in the chat. It was fantastic. I would like to thank you. I would like to thank all the other speakers of this morning for this very interesting session. I learned a lot, and I will contact some of you again to find out more about the details. I would like to thank once more the organizers, especially Kazimierz. Thank you very much for letting us participate. And I now hand over back to you, Kazimierz.
Thank you, Horst. Thank you, all participants. Thank you, Mark. But this was the last presentation of today's session devoted to health technologies. I would like to remind you that there is another one partner from this consortium, C4BIC, which aims the consortium which aims to create European partnership for personalized medicine, and we'll be working together in this project for the next year, and after this year, we'll hope to have these partnerships established and running on. Tomorrow, I want to invite you to the next session of personalized medicine or health technologies, which will be led by third partner or fourth partner, or in that case, I would say first partner, because the Atlantopol Therapies is the leader of this project. For now, I thank you very much. We terminate the session right now. You've been visiting the Krakow, Life Science Krakow. See you later.
Now the turn is on. Thank you once more, Kazimierz. Take care. Have a good conference. Bye-bye.